Well, why don't we start? So it's my pleasure to introduce our seminar speaker today. Um, Dr. Scumpia is at UCLA and the Division of Dermatology in the Department of Medicine. He is here at UCI, kind of exploring the Department of the Division of Dermatology here. And many of the people on this call, or on the Zoom call, are very interested in skin. And the title of Phil's talk is Translating an Understanding of the Cutaneous Microenvironment into Diagnostics and Treatments for Wounds, Cancers, and Inflammatory Dermatoses. So Philip, go ahead and take it away.
Thank you, Dr. Milner. Can everyone hear me all right?
Yes.
Okay, good. And my screen share is going okay. I'm going to turn on the presentation. Do you guys see it? See a normal presentation? Okay, good.
Yeah, it's full screen.
Sometimes it goes into presentation mode and you see multiple screens.
Yeah.
Okay.
You just see the one screen.
Perfect. Okay, great. So thank you guys so much for having me. I really appreciate being invited to come and give a talk. Now, I had trouble finding a title for the talk because really I just wanted to give you guys an overview of the type of research I do. And, you know, I do wound healing research, I do some cancer research, and I do some inflammation research. I also do some non-invasive diagnostic research that's more clinical translational, that is kind of because of my background in dermatology and dermatopathology. And unfortunately, because there's some patents pending, I was told by UCLA, I am not allowed to discuss anything more than just a general idea. So I'm not going to be able to actually go into those projects, but I'll mention what I'm doing in the diagnostic field, and then I'll focus most of the time on, sorry, there's a guy with a blower that's about to go and it's going to be really loud in a second. But basically, I'll talk mostly about my basic science research. So as I mentioned, I have patents and patents pending regarding several of the projects I'll be discussing. I'm a co-founder and shareholder in Tempo Therapeutics, a company whose goal is to commercialize the hydrogel that I'll be discussing in the talk. And I unfortunately can't describe some of the cool diagnostic projects that I'll wanted to discuss because UCLA told me I am not allowed currently. So a little background, so an outline of my presentation, I'll give a little background and then talk briefly about my research theme in minimally invasive diagnostics. And then I'll spend the majority of my time talking about the other research theme that I have. How can we manipulate the cutaneous microenvironment to treat, to learn about skin disease, to develop new treatments? And I'll talk about some future directions of my work as well. So first, a little background. And I just wanted to kind of go over my approach to some of the research projects that I do. My goal is to really try to help understand and treat complex skin problems, and I'll go into my approach into the various topics. So the first topic is the development of minimally invasive skin diagnostics, and basically how I do both on the dermatology and dermatopathology end. We do skin biopsy. So you either you anesthetize the skin, you clean it off, and then you either take a punch of the skin, you cut it out, or you shave it off. That tissue gets fixed, processed, and put on microscope slides, and then you get histologic images. We've been doing this since the 1880s, and really there hasn't been that much innovation until the past 30 years. despite there being multiple innovations in CT scan, MRI, the more invasive imaging, which has really helped prevent unneeded biopsies. So what are some ways we can do skin biopsies, quote unquote, or skin imaging and getting a skin diagnosis without cutting and removing the tissue? So one approach is optical biopsy. And the Beckman Laser Institute is very familiar with probably all of these, but there's reflectance confocal microscopy, which is one of the oldest. There's multiphoton microscopy, optical coherence tomography, Raman spectroscopy, and the list goes on and on. There's lots of different approaches that are being investigated as well. So while the approaches don't require tissue biopsy, they do require someone to interpret the images, a diagnostician who's familiar and skilled with reading the images from the procedure, because all of the images, they look different than histology. So a lot of these imaging techniques haven't really taken off, even though they're clinically approved devices for their use, because it's challenging to read to get a diagnostic diagnosis, basically. So my approach in one of the projects was, is there a way we can transform the images that we see to get histology quality images using an optical biopsy technique? And in the project, basically what the goal is, we took an RCM image of skin, and how it looks is this black and white image. And what we do is we transform it into something that looks like histology. I can't unfortunately describe how we do it, but it's a really cool study. It's taken just over a year to do. And what's really neat is we can see some basal cell histology that looks similar to what we see under the microscope. And another approach that I'm using is more molecular biopsy. So in molecular, so how molecular biopsy works is you get a molecular signature or a gene mutation, and that's been around for a long time in pathology. So FISH, CGH, or comprehensive genomics hybridization, those have been used for many years now to look and see if specific mutations, or more broadly, several mutations, are present in tissue. It's not really made for diagnosis, but it's made to help with just seeing what the mutation is for treatment approaches. Some newer companies have started using molecular biopsy, where they take a piece of skin, or in the case of DermTech, they have a pigmented lesion assay where they take basically an adhesive tape that they put on the skin, let it sit for a minute and remove it. And then that gets converted to RNA, and the RNA, they look at 3 genes, and that gives you an answer. There's other ones, Myriad MyPath, which is similar to the DermTech pigmented lesion assay, but it requires tissue. And then there's Decision DX from Castle Biosciences. Those require actual tissue, so they don't really save the biopsy as compared to the DermTech lesion. So they claim that they can get results just as accurate as skin biopsy for determining the difference between melanoma and a nevus, whether it's atypical or a benign nevus. And this only works in the case of the assay, because melanoma, if you look at the histology, this is the histology of skin. What happens is melanocytes, when they're malignant, instead of only going down, they also go up. We call it pagetoid spread. So when they put the adhesive on the skin for melanoma, you can detect some of the RNAs from the melanocytes that are closer to the surface. And that's how the sensitivity of their assay works, because in a benign lesion, you don't see the melanocytes at the surface. So if you detect those melanocyte genes at the surface, it's more likely to be malignant than not. So how can we develop something similar for different types of skin disorders? And as a dermatopathologist, besides determining if something borderline is melanoma or is it an atypical nevus, another bane of my existence is inflammatory skin diseases, because many times the clinician does the biopsy and they say, rule out psoriasis or eczema. And when I get the biopsy, I'd say about half of the cases, I can tell them this is either eczema or psoriasis. And I can't give them any more information than that, because they're so difficult. If an inflammatory disorder has been going on for long enough, it's very difficult to tease them apart and say which one it is. So dermatopathologists often hedge, and we can say, well, we favor eczema in this case, or we favor psoriasis in this case, but we can't tell for sure. So the approach that we wanted to use is basically, rather than just using an adhesive to get the surface RNAs, we need to get into the dermis where the inflammatory cells are as well. And the reason being, I was involved in a study several years ago where we looked at microarray assays from over 15 different skin conditions. And what we found is we can segregate the diseases based on the gene expression profile very well. So our goal in this project, and this is a collaboration with a couple of bioengineers at UCLA, Dino DiCarlo and Song Lee, what we want to do is make microneedle patches that can test various depths, test the RNA, collect the RNA from various depths. Then we get the nucleic acids and we can process it using a rapid test based on genes that are expressed for different inflammatory dermatoses. And there's a potential to get the answer very rapidly in clinic, even if the LAMP PCR machine, the LAMP or PCR machine is in the office, or you can send it for centralized testing. So again, I can't go into this project very much, but because of pending patents, so I can't tell you how we're doing it. But maybe in the future, I'll be able to do that as well. So those were kind of my two projects on non-invasive skin diagnoses. And I'm really excited about them, but unfortunately, I can't talk more about them. Where we're going from there, so we used our first approach to actually get histology quality images of the skin. We did it for normal skin and basal cell carcinoma. Now we need to expand to see if it works on different skin conditions, such as squamous cell carcinoma and seborrheic keratosis. The reason being, those are a little bit challenging because they're thicker, and RCM imaging is, it only gets the very surface, so it might not be able to detect it quite as well. Also, we have done pigmented lesions. We've done nevi, but we haven't done melanoma. So the key is to see if we can detect differences between nevi and melanoma there. And obviously with reflectance confocal microscopy, how we get the images, they're in the horizontal plane, which nobody in pathology is used to. To get people more used to it, we'll need to do some 3D reconstructions or transformations to transform it into the vertical plane, which is what pathologists are used to. So the goal will be to apply that approach to obtain the histology quality images. And then for the future directions of the molecular biopsy. The goal is to see if the signatures that we identify really translate to what we see. So we took stereotypical psoriasis, stereotypical atopic dermatitis, but will it work in those conditions that are the tweeners on pathology? Will we also be able to detect the differences in the molecular profile to tell whether something that's on borderline by histology and clinical to see the difference in the molecular signature? So now I'll switch gears to my other projects, which this is my research themes kind of in the basic science realm of skin disorders. So what I aim to do is basically identify how a disease is occurring and basically try to manipulate either the cells or the microenvironment in the cells to be able to see how it impacts the disease, whether it's inflammation like psoriasis or eczema, an infectious disease like staph or strep infection, or a malignant skin disease like melanoma. So why do I care about the skin besides the fact that I'm a dermatologist and a dermatopathologist? Well, it's the largest organ in the body. It's unique and complex. There's many structures that regulate the barrier of the skin. The barrier of the body. It also regulates temperature, how we sweat, how our blood vessels open and close can regulate temperature. It can store fuel with the fat in the adipose tissue that's below the skin. And also it responds to stresses. It's very elastic, it's movable, it's durable, and it helps to basically, when you do undergo trauma, to prevent it from breaking the skin. But then when it eventually breaks down, we have to be able to repair it. So it mediates the responses to that. The other thing, I'm also an immunologist, and the skin is one of the most strongly immunologic organs in the body. There's sentinel cells, including Langerhans cells, dendritic cells, macrophages, mast cells, at every level of the skin. These sentinel cells and stromal cells are primed for rapid responses so that they can recruit other cells to the skin so that they can respond to either a pathogen or a breach in the barrier or a cancer or UV light. And in theory, stromal cells are immune privileged, but eventually that can break down as well, like what happens in response to an allergy or Steven Johnson, when your own immune system attacks the skin. So it's also very complex because we have connective tissue in addition to the cells that make up the epidermis, the cells that make up the dermis, the immune cells, the hair follicles, and other structures. We also have collagen, glycosaminoglycans, hormones, and neural signals. And all of these contribute. So we have neural signals that regulate fibers, that regulate itch, that regulate pressure, mechanical disruption, and even stress. So stressful events can make our hair stand up. So all of these things make the skin such a unique environment for us to respond to to changes in that environment. So how does it do that? It does it by integrating signals from the local microenvironment, but then also bring in other cells to respond to the stimuli and maintain homeostasis. So I developed a quick model of how I think about an injury to the skin, whether it's a wound, a cancer, UV, or an inflammatory signal. So what happens is typically you get a stereotypical response by the sentinel cells, the cells of the skin, and immune cells. And then you get a stereotypical response from the nervous system, from hormones, and even from the extracellular matrix, so that you can get, for instance, a fibrin clot so that you stop bleeding. So all of these things are programmed evolutionarily. So you get this evolutionary pre-programmed biologic response. And typically the evolutionary pre-programmed response is, if you get wound, you get a scar. If you have cancer, you try to resist the cancer, you try to attack it, but eventually the cancer grows because of immune evasion. And then if you get an inflammatory signal, you can get a dermatitis. So all of these factors contribute to these stereotypical pre-programmed responses. So my goal is, can we come up with a way to directly alter either immune cells, keratinocytes, or fibroblast responses, or to transform that pre-programmed response to get skin regeneration rather than skin scarring, to get a tumor to regress rather than to get the tumor to grow, and to prevent or reverse a dermatitis, whether it's psoriasis or eczema, to get back to more homeostasis than what's previously possible. Or there's another way to do it, which, can we manipulate the nervous system or hormonal cues or ECM cues, extracellular matrix cues, to indirectly alter the immune response, keratinocytes and fibroblasts, and get that same transformed response? Basically, get rid of that pre-programmed response into something that's more favorable. So that is kind of what I'll talk about. I'll talk about two projects, how I went to do it. One of them is in wound healing, and one of them is in cancer. We tried it in several different cancers, but the main focus was melanoma, or just an aggressive skin cancer. So in the first project, we wanted to see, is there a way that we can give the dermis, rather than that stereotypical response where you get the granulation tissue and that goes to fibroplasia, and then that fibroplasia eventually gets replaced with a scar tissue? Can we put a synthetic dermis there that can mimic what was there before, so that we can heal the wounds better? And to do this, we have to understand how the skin is at baseline. So, we have the epidermis here, which is just filled with keratinocytes. There's really no space between them. But when we look at the dermis, the extracellular matrix is actually very porous. And it's basically collagen bundles, elastin tissue, and then all of these water-bearing molecules like glycosaminoglycans, so that cells can go in there rapidly, see what is going on, and if it needs to, they can degrade it. So our hypothesis was, if we can make a synthetic dermis that has all of the cues of a real dermis, and make it degradable, so that eventually when the cells fill it in, because it's porous, that we can recreate a more normal skin and improve wound healing. So this was published back in 2015. Basically, what we did is we used microfluidics, and we pumped hydrogel and peptides into little beads. We made little beads of hydrogel that have, on the outside of them, they have different peptides, K and Q, which mediate an annealing reaction. So each polyethylene glycol molecule has 4 arms, and at the ends of them, you have different peptides, whether it's K or Q. And on the outside of the PEG molecules, we also have RGD, which is an integrin binding protein that can mimic the extracellular matrix, because RGD is also present on collagen. So what happens is, In response to factor 13 or thrombin, factor 13 and thrombin, or white light and eosin Y, we can get these particles, which are a liquid initially, we can get them to anneal once you put them inside the tissue. And what happens is you can flow these beads, and basically they anneal, and you have little spaces that develop between the beads so that cells can grow into. And we used it in an excisional wound model, and we splinted the wounds so that the wounds didn't just heal by contracture. And what we see is when you have a non-porous gel, one that's how people have been using hydrogels for many years, what happens is you get a foreign body reaction, and you get immune cells that build up sort of like a splinter trying to push this hydrogel material out. But when you have spaces between the hydrogel particles, the cells can flow in between them. And in fact, you can kind of see blood vessels starting to form even within 3 days of having the hydrogel there. And what we found is when you use a MAP scaffold compared to the non-porous hydrogel, we find an improvement in the wound closure at 7 days following the wounding. So we thought this was unique. We thought it was cool. So we wanted to study it further. The reason being, the base material is completely tunable. We can change the properties how we see fit by changing the peptides, by changing the annealing mechanism. And what happens is we can see if the response we get can improve outcome. So the first experiment we tried to do is we wanted to see if we can make the hydrogel stick around longer. So what we noticed is when we put the hydrogel into the wounds, what would happen is after about 7 to 10 days, the individual hydrogel particles, because they get degraded by the body's natural MMP system. So MMP, the peptides have a degradable sequence where they connect to other hydrogel particles. And what happens is if MMPs are present, it cleaves the hydrogel particles, and then each particle can get further broken down by MMPs. So we wanted to see if we changed those MMP particles, the MMP peptides, from the L chirality to the D chirality, basically the mirror image, MMP, they should be more resistant to MMPs, and they should degrade slower. And when we tested it in vitro, what we found is the material properties of the hydrogel were the same, but what happened is in vitro, the hydrogel particles took much longer to degrade when the D-chiral peptides were used. So we thought this could be an interesting approach to prolong the hydrogel from staying there. But what in fact happened when we used that same splinted wound model is the hydrogel actually degraded more quickly. And this was a paper we recently published in Nature Materials. But what was interesting is the hydrogel was gone much quicker. But what was interesting is in the normal untreated wounds or wounds treated with a normal hydrogel, this is looking 21 days later. So after the wounds have completely healed and the hydrogel is gone, we have a scar when we don't have the D-chiral amino acid peptides. So you see flattening of the reedy ridges, you don't see any hair follicles forming, you don't see any sebaceous glands forming. And that's what we see in a typical human scar as well. However, when we look at the DMAP hydrogel, the ones treated with the D-chiral peptides cross-linking the hydrogel, we saw numerous hair follicles forming within the wounds. The hair follicles seem to be forming from these cystic structures, so they were forming from follicular cysts, and we saw more sebaceous glands as well. So when we looked at why this was occurring, when we looked earlier on, we noticed that the DMAP hydrogel had more immune cells present in the wound. So we were thinking that the DMAP was eliciting an immune response somehow. So first, we wanted to see if the D-peptides themselves were doing something funny. So D-amino acids, D-peptides have been shown to activate the FMLP receptor, basically FPR2, which is kind of a G protein coupled receptor that can activate immune cells to produce inflammatory molecules, and it's also chemotactic. We thought maybe that the D peptides were directly activating pathogen-associated molecular patterns. However, when we treated macrophages with super high concentrations of the D peptides, 100-fold higher than what was seen in our hydrogel, we saw no macrophage activation, very similar to the L-amino acid peptides. And they also didn't boost the response of macrophages by low-dose LPS. So we were pretty confident that they weren't activating the innate immune system directly. So what we thought was perhaps in the context of the MAP gel was allowing immune cells to infiltrate. And then when the immune cells were breaking down the hydrogel particles, they were taking the D peptides, which were acting as an antigen. So the MAP gel was acting like an adjuvant, but not an adjuvant like what we would typically see with a toll-like receptor agonist or a STING agonist per se. So a paper came out in Nature Materials while we were working on this project. And what they found were in joint replacement, there's metal and plastic particles that form because of the shear stress on the joints. And those small metal particles from those implants, those microparticles, can directly activate macrophages to produce interleukin 33. And they found it required Bruton's tyrosine kinase and Syk kinase. If you block those, the macrophage wouldn't elicit this type 2 immune response and induce interleukin 33. What was interesting is they found a type 2 immune response. So there were a lot of T helper 2 cells in the macrophages that were activated by those particles. However, their microparticles were completely non-degradable. So what my theory was, because our hydrogel particle does eventually degrade, even with the D-amino acid peptides in it, and it probably degrades more rapidly because of non-specific hydrolysis. So the peptides are filled with water, and the hydrogel particles get degraded by just oxidative stress produced by the immune cells. Once we get rid of those signals, because the amino acid hydrogel is gone, instead of causing fibrosis and what they found in the non-degradable microparticles, we found regeneration. So an acute burst of these Th2 or M2 pathways elicits a regenerative response, whereas if it becomes chronic, it can also be detrimental. So to test this, what we did is we put subcutaneous implants in mice of L-amino acid hydrogel or D-amino acid hydrogel. And what we found is we did in fact see IL-33 expressing macrophages in both D and L-amino acid hydrogel. In fact, the majority of the cells, over 90% of the cells in our hydrogel in these implants 21 days later were macrophages. And of them, a large number of them produced IL-33. So if you look at the edge of the implant, so this is kind of the outside and this is the center of the hydrogel. what we found is there were cells in the middle of both hydrogel, but they were much fewer than at the edge. And we saw a robust immune response. So if you look at the core, there really was no difference in macrophages in the D amino acid or L amino acid hydrogel. But when you look at the edge, there was a threefold increase of IL-33 producing macrophages in our hydrogel implants. So we wanted to now see, are we in fact eliciting an adaptive immune response to the D amino acid peptides? And the answer is yes. So mice that received the L amino acid hydrogel did not make IgG1 antibodies, they didn't make IgG2A antibodies, and they didn't make IgG3 antibodies, which are associated with Th2 immune responses, Th1 immune responses, or T cell independent immune responses. These are mice that were injected with L hydrogel only. Now, when we looked at the mice that were injected with D hydrogel, what we found is there was a Th2 type IgG1 antibody response that was produced. There was some mice did get a small IgG2A or Th1 type antibody response, while really a few mice, again, non-significant, got an IgG3 type T cell independent immune response. And again, that was non-significant as well. So we wanted to prove that the adaptive immune system was necessary for the regeneration. And what I'm showing here are just B6 mice that were given either LMAP or DMAP. And what we find is the DMAP treated mice did have hair follicles. However, when we got rid of the adaptive immune system, we did not see regeneration in sham mice or in the DMAP treated mice. So if you got rid of the adaptive immune system, we did not see the hair follicle regeneration. And just another example of the DMAP, you can see the DMAP particles with hair follicles forming directly over the hydrogel. And what we see is only the DMAP treated wild type mice had hair follicle formation. So we proved the adaptive immune system was required for the hair follicle regeneration. So where do we go from here? So we know that proper activation of immune cells within the microenvironment can allow for regenerative wound healing. And the unique properties of the hydrogel can make it a promising biomaterial if you want to directly activate immune cells within a microenvironment. So a really cool direction where we're going with this project is, could we use the implants as a vaccine? So could we use it as a vaccine platform where you get slow release of the antigen? And basically, what would be really cool is you could, since it's individual hydrogel beads, you can kind of think of this off-the-shelf like library approach where you have different antigens, you have different nanoparticles that you want to release if you want to convert an immune response from Th2 to Th1. And you can do that all within one vaccine because each blob of hydrogel is its own unique entity, and you can combine it before you inject it into the person. So basically, in proof of concept studies, we're calling it VaxMap, or vaccine map hydrogel. Basically, what we wanted to do is, can we tune the immune response by adding something to it? So here we immunized mice with nitrophenylalbumin, a standard immunological antigen that's been used for many years. And we did it compared to complete sporoid adjuvant, alum, and then here's the VaxMap without CPG or with CPG nanoparticles, and compared to mice that received NP-OVA in PBS. So what we noticed is if we look at the total IgG or the IgG1, the Th2 antibody responses, we see that the VaxMap elicited, the slow release of the VaxMap elicited a response of total IgG and IgG1, the Th2 type, very similar to CFA and slightly better than alum. When we added CpG to it, we saw similar levels. But what was really cool is we were able to tune the immune response by adding CpG. We were able to get Th1 type antibody responses to NP-OVA. And what I'm showing here is the high affinity antibody responses to NP. So the cool part about this system is you can measure low affinity and high affinity antibodies, with obviously high affinity antibodies being better. And basically more, that means that the B cells have affinity matured. When we looked at the low affinity antibodies, they were actually similar between all the groups, whether it was CFA, alum, CpG, and CpG, with a VaxMap without and with CpG. But for total IgG and IgG1, and only CFA and CpG elicited a strong low affinity IgG2C Th1 response. So next, what we wanted to do is we wanted to see, can we do it with an antigen that's relevant to something, I don't know, something that's going around these days, maybe COVID. So what we did is we loaded the MAP particles with the spike protein, and we immunized mice, either given alum or VaxMap. This was a combination that we were trying to dilute out the amount of the spike protein by giving a lower dose to see if it played a role. So it was the same concentration, but only in half of the particles. And what we found is, looking at serial dilutions, we find that total IgG and IgG1 responses are very similar and very strong to RBD. So we're getting anti-RBD, or the neutralizing component of the spike antigen, in these immunized mice. We didn't make much IgG2C or the Th1 type antibody response. So the key now is, how can we incorporate two antigens? And can we incorporate two antigens? I know at the beginning of the pandemic, there were lots of people talking about antibody-dependent cytotoxicity, because you're getting too much of a robust immune response. So what we were thinking was, in response to a different antigen where you don't want to get a strong antibody response, could you drive the antigen intracellularly in the macrophages or dendritic cells and allow for cross-presentation, so you just get a CD8 T cell response. So what we're doing is we're trying to load some particles with the N protein versus some particles with the S protein. And the N protein is going to be loaded in mesoporic silica nanoparticles that we put into the MAP hydrogel. And the goal will be to combine that into one vaccine and see, do we get strong neutralizing antibodies to the spike protein, but get less antibody produced to the N protein, which would not be neutralizing, but we get activation of cytotoxic T cells. And since N protein is fairly homologous among, about 80% homologous among different strains of coronaviruses, you could imagine you'll get T cell responses against multiple strains of coronaviruses that have similar N proteins. And then finally, could we use this as a sort of cancer vaccine where we're injecting MAP hydrogel with different either antibodies that are checkpoint inhibitor antibodies or medicines directly into tumors to get an immunotherapy boost. So the final project that I'll talk to you guys about is how I was thinking about tumor cells and microenvironment and how they can contribute to anti-tumor immunity. So in 2017, a talented postdoc in the lab, we published a manuscript looking at how ionizing radiation activates different pathways in macrophages. But to simplify it, what we found was basically ionizing radiation activated ATM to directly activate IRF1. And this allowed for type 1 interferon to be produced. So type 1 interferon is important for a lot of cells in the tumor microenvironment. It's produced by macrophages and dendritic cells, and it activates NK cells, cytotoxic T cells, and that can elicit some tumor rejection. And right around the same time, the Rebus lab found that IRF1 controlled PD-L1 expression in melanoma cells downstream of interferon gamma production. And what was really interesting, that non-invasive skin diagnostic, Myriad MyPath, they actually have IRF1 as their signature gene to tell if something's melanoma or not. So we thought we were on the right path, and we wanted to see whether IRF1 plays a role in the tumor microenvironment or in the tumor cells themselves. So to do this, what we did is we looked at IRF1, basically, we looked at IRF1 knockout mice and wild type tumor cells to see if the microenvironment of the mouse, if IRF1 was playing a role there. And what we found is basically in IRF1 knockout mice, if you give MC38 tumors, YUM melanoma tumors, or B16 melanoma tumors, IRF1 knockout mice have an astounding growth of tumor compared to wild type mice. And it was even greater than STING knockout mice. We found that basically IRF1 knockout mice can't recruit cytotoxic T cells or NK cells into the tumor. So IRF1 in the host basically controls the development of CD8 and NK cells, but also they can't proliferate as much locally as well. So with these defects, the tumor grew very large. So next, what we wanted to do is we wanted to mutate the tumor cells to get rid of IRF1. And again, we chose the MC38, the B16, and the YUM line because of the differences in immunogenicity and tumor mutation burden and MHC expression. And what we did is we knocked out IRF1 using the CRISPR-Cas9 system. And we also knocked out STING as a control. And in vitro, those IRF1 deficient tumor cells grow, proliferate just like wild type tumor cells. But what was really interesting is after a short period of growth, the IRF1 knockout tumors regressed completely. And that was completely different than wild type tumors and STING tumors when we used MC38 cells. So when we look at the tumor weights, they're completely gone. Only one mouse had any tumor growth. We also found the same thing in B16 melanoma cells and YUM tumor cells. So we didn't even have to give an immunotherapy PD-1 inhibitor or a CTLA-4 inhibitor for these tumor cells to shrink. Next, what we wanted to do is we wanted to see if this was immunological regression. So what we did is we gave the mice to we gave the wild type versus IRF1 out MC38 cells to basically rag gamma C mice that have no adaptive immune system, or innate lymphoid cells, including NK and NKT cells as well, along with NOD-SCID mice. And tumor growth in both of these strains was very similar to wild type mice. So we know that the regression we were seeing was because of the effects on the immune system. So we wanted to look further at it. Was it NK cells or cytotoxic T cells? And what we found is the IRF1 deficient tumor cells, recruited more CD3 cells, CD4, CD8, and NK cells. And when we look at the tumor, you can see both inside the tumor and at the tumor margin of wild type mice, there were few immune cells. But when you look at IRF1 knockout mice, CD4 and CD8 lymphocytes were much more enriched, and there was much more tumor necrosis in the IRF1 deficient MC38 cells. So when we looked at rag mice, which still have NK cells, and CD8 knockout mice, we found a partial restoration of tumor growth. So both NK cells and CD8 T cells were contributing to the tumor growth when we compare it to the wild type mice or the NOD-SCID mice, which had similar tumor growth. So what could be the mechanism of this? So we looked downstream, and obviously because of the Rebus paper, we were thinking that maybe it was PD-L1. And PD-L1, if you give interferon gamma to these IRF1 deficient mice, they can't upregulate. So the expression goes up 20-fold in the wild type mice, but it doesn't increase in the IRF1 mutant mice, whether you're looking at MC38, B16, or YUM cells. So we got rid of PD-L1 in the IRF1. We overexpressed PD-L1 in the IRF1 deficient tumor cells, but that only partially restored the growth. So some of the tumors did grow when we overexpressed PD-L1. So that told us that it wasn't only PD-L1 that IRF1 was regulating. It also regulates a few other candidates, including TRAIL. So TRAIL is another factor that is diminished in the knockout mice. And what we found is that basically there's a group of genes that are dependent on IRF1, but then genes like FAST that regulate cell death are actually overexpressed in IRF1 knockout cells. So it could be contributing to cell death pathways. So we have to figure out how that's happening. So basically future directions is we're going to do the same thing with TRAIL that we did with PD-L1, and the same thing with FAST. We'll knock out FAST to see if that affects the tumor growth and see how IRF1 is regulating tumor growth. And also in the future, we found that there's a strong lipid signature in the tumor cells. So lipids go down as the tumor gets attacked by the immune system. So we want to see, is IRF1 regulating lipid metabolism? And then could we use our hydrogel to deliver something like siRNAs to decrease IRF1 expression, and could that be used as a cancer vaccine? And then obviously the more mechanistic studies is how is IRF1 regulating PD-L1 and TRAIL, and how is that regulating the tumor growth? And we think that IRF1 in cancer cells controls how the tumors can protect themselves from the anti-tumor effects of interferons in T cell and NK cell recruitment. So how is IRF1 in immune cells regulating the tumor microenvironment? Well, we know it regulates the development of cytotoxic T cells and NK cells. And it also regulates interferon gamma production downstream of type 1 interferon. So if you give type 1 interferon in a tumor, it'll regulate interferon gamma to bring in more cytotoxic T cells and NK cells. But it'll also regulate PD-L1 expression and other factors which could diminish that. So we have to find how the yin and yang is occurring between the immune cells and the tumor cells. So with that, I would like to thank all my lab members who Prabhat Purby was mainly in charge of the cancer project. Li Jing was in charge of a lot of the wound healing project. Ariel Floro did a lot of the work with the microneedle patch. And Mani Archang did a lot of the work with the hydrogel. And obviously I couldn't have done any of this without my collaborators, Dino DiCarlo with the hydrogel work, Jason Weinstein with the vaccine work, and Max Plikis here at UCLA with the hair follicle regeneration work. He was critical with that. And I would like to thank my mentor, Steve Snail, for helping me find the lab and my funding sources, my KO8 and my RO3 from NIAMS, and a cancer center grant from the JCCC. So with that, I'll take any questions, and thank you all very much.
Thank you. That was excellent. Questions?
Can I ask a quick question, because I need to run? That was a fantastic talk. Thank you. I'll see you later too. I'll ask more questions then. But just a quick one from earlier in your talk. You were talking about that microneedle patch. Is there any concern about poking into a potential melanoma lesion with a microneedle?
It's possible. So typically it only goes up to 900 microns, which is the very superficial part of the dermis. I mean, the other thing, when you do poke it, there's a chance you can cause micro injury, which will bring in an immune response. So there's also the chance that you can bring in immune cells to help with control of the tumor as well. I mean, it is a possibility, and that's partly why we're going with inflammatory dermatoses first, right? And you know, we're not going for you know, melanoma, because there's already an assay that works without the microneedle. Mm-hmm. So with just adhesive.
Yeah. So I see. Yeah. Okay. That was great. I'd love to. I'll talk to you later. I'm looking forward to it. Just need to run into another meeting now. Thank you very much.
Sorry for going over.
No problem. Thanks.
Dr. Perlman.
Yeah. Hi, Phil. That was tour de force for sure. It was really excellent work all around. I had a couple of questions with regard to this as possibly using this as a possible adjuvant. You know, your antibody titers were really impressive, you know, right on par with complete Freund's adjuvant, which of course you can't use in people. But I was wondering, well, two questions. One is, have you had a chance to see if the anti-spike proteins, the antibodies to the spike proteins, are they actually neutralizing? Do you have a setup there to do that?
Yeah, so we do have a setup to do it. The problem is we use cell culture that requires a lot more of the serum and than we can get from the mice. So we need like 20 microliters, and that we've only done tail bleeds on the mice so far because we want to see over time. Yeah. That experiment just started 2 weeks ago. And that was 3 weeks ago. And 1 week ago was when we got that blood at day 10. So we want to see the response over time, but at the terminal bleeds, we do want to look at live virus neutralization to see if it neutralizes. Right now, for the small amount that we get from the tail bleeds, we are only looking at anti-RBD antibodies and total anti-spike antibodies.
Well, we do
as a
have,
surrogate.
we have 2 BSL-3 labs here at UCI, and COVID research is being done in both of them. So, you know, that's something to consider as well. Do you see any damage to the tissue when you're giving the vaccines? Is there any reactogenicity, I think it's called?
Yeah, so what it does is it causes a bubble. And it's like, do you know what Juvederm or Restylane, like hydrogels are? No, so they're basically tissue filler people use to, you know, make people prettier. And basically what we see is something very similar to that, except we see cells that infiltrate. Eventually it goes away. And within like 2 months, the hydrogel is completely gone.
Oh, really?
So it does form a lump. So that's the one caveat, right? You get a bump because it's a little hydrogel under the skin, but it deflates because of your immune response. Now we can tune it, because in another project, we're actually looking to see if we could be a next generation tissue filler as well. Because of the cell infiltration, could you help rebuild aged tissue? So that's a separate project. And you can use it to deliver cells. So if you want to deliver stem cells to wounds, or deliver, CAR T cells to a tumor, that's like pancreatic cancer, that's, you know, immune poor, right? That could be a way to attract cells to that tumor.
Great, thanks. It's exciting. Okay, so I have one more quick question.
Go ahead.
In the tumor studies you were doing, you showed, I mean, it was really impressive to see the T cell infiltrate around the IRF1 knockout tumors. And you're wondering what part of the mechanisms were, but I mean, just a quick glance, you've got some upregulation of chemokines there as well, T cell chemokines. Is this something you've been considering looking at? And I'll bet there's other chemokines that are upregulated as well.
Yeah, so CXCL9, sorry I had to fly through that part of the talk. CXCL9, 10, and 11 are part of the interferon response that's not regulated by IRF1. So if you knock out IRF1, you're losing trail, you're losing, you're increasing FAS, and you're also decreasing PDL1, but you're able to attract T cells through CXCL9, 10, and 11.
Yeah, that makes sense.
So you're getting all the good effects of interferon, but blocking a lot of the bad effects. I also didn't go into MHC2 and 1 expression are regulated by interferons through IRF1 as well. It's not fully through IRF1, it still goes up, but at baseline, there's no MHCs that are expressed in the IRF1 knockout. So it's multifactorial. And it all adds up.
Yeah, that's really exciting work.
Thank you.
Thanks, Philip.
Thank you.
Other questions for Phil? I have a couple questions, Phil, if you have time.
Sure.
So, you know, at the very beginning of your talk, when you talked about the non-invasive diagnostics, and you presented
Hold on.
Yeah. Hey, it's me. Maybe this will be the last question though, because he's got a little bit of a tight schedule. We just have a few minutes to eat, and we've got to tour a bunch of places. So I do apologize. So we'll
okay.
Last one, and we'll wrap up from there. Yeah, I'm sorry. Thank you.
Okay. Yeah, the imaging methodology. Can you say, I know you have some IP stuff going on, but can you say about what the size of the field of view of the optical approach that you developed?
It's the Viviscope that we're using.
Oh,
So
okay.
it's the same field of view that the Viviscope has.
Okay.
I think it's like 600 by 600 microns.
Okay. Yeah, I guess, I don't know if we have time, but you know, the second approach using the microneedles, I'm just wondering kind of what your view is on the optical versus these approaches like microneedles, where you're actually harvesting material and then can do things with the material, which seems to have some really important advantages over what you can do optically.
Yeah, I think that the combination is where I see the future of diagnostics, right? I think the optical biopsies are getting incredible, especially now with multiphoton microscopy. And if we can convert that into something that looks like H&E, I think that would be great. But even that, it doesn't tell us the whole picture, right? Same thing with, that's why the molecular approaches can give us images that can give us insights that we can't get just with images. And that's why I need, as a dermatopathologist, I need special stains, immunostains, and even then sometimes I can't get to the right diagnosis. So I think doing these combination approaches will eventually be the state of the art where all, diagnostics are heading.
Okay, well, thank you very much. I know you're on a tight schedule, so we really appreciate your work and your talk.
Thank you guys so much. I really appreciate you listening.
Okay.
Bye-bye.
Bye-bye.